Hi everyone, and welcome to the Chapter 10 vocab video for Chemistry 100. This chapter is all about proteins. So all of these words have to do with proteins or enzymes. So the first word, active site, is referring to the site on an enzyme where the substrate binds to. Substrate is another word. A substrate is a thing that binds to an enzyme, and it binds specifically at the active site of the enzyme. It's where the catalysis occurs. An alpha helix is a shape that a protein can take. It's part of the secondary structure of a protein. So it's a sort of, think of it as like a telephone cord coil. That's an alpha helix shape. Not to be confused with a double helix, which is the shape of DNA. Amino acid is a building block of protein. So it's the monomer, the single unit, and we string them together to make a polymer of amino acids, and we call that a polypeptide, another one of your words. A polypeptide is a string of amino acids that are connected by peptide bonds. Peptide bonds hold amino acids together. Beta pleated sheet is another shape in the secondary structure of a protein. It's sort of a folded back and forth shape. Sometimes it's sort of equated to like a paper fan. Conformational change is a change in shape that a protein can take. And proteins will sometimes undergo conformational changes when a substance binds to them, or if they're put in a different environment of a different pH. So it's a change in conformation of shape. Coenzyme and cofactor, those are things that bind to enzymes to activate them. So a lot of enzymes won't work without the necessary coenzyme or cofactor. The difference between the two is small. A coenzyme is an organic molecule. Typically they are things that we refer to as vitamins. A lot of the vitamins in our diet are actually important coenzymes for enzymes in our bodies, and cofactors are usually ions. They're usually metal elements, so they're very small, single atoms or single ions, versus coenzymes, which are slightly larger organic molecules. There's different types of inhibitors. An inhibitor is something that stops an enzyme from working. It prevents it from working. A competitive inhibitor is one that binds at the active site. It competes with the substrate, and it oftentimes looks a lot like the substrate, mimics the substrate. A non-competitive inhibitor is one that binds somewhere else on the enzyme, often leading to a conformational change that then the active site won't fit the substrate and the enzyme can't work. Let's see, where were we? Denaturation. Denaturation is a chemistry term for what happens when a protein becomes unfolded. So I've already mentioned the secondary structure of proteins. Proteins have 4 layers of structure: primary, secondary, tertiary, and quaternary. So they fold up into this really tight 3-dimensional shape. And certain things like acids and bases and alcohols and heat can all cause the protein to denature or to unfold. So it doesn't break peptide bonds. It's not that the protein is breaking, it's just unfolding. And it won't work if it's unfolded. So we call it denatured. A denatured protein is one that is unfolded. Sometimes, in some cases with proteins, you can renature them or refold them, but sometimes once they're denatured, they're permanently denatured. A dipeptide is 2 amino acids bonded together. Like a tripeptide would be 3 amino acids bonded together. A disulfide bond is a type of bond that occurs between 2 sulfurs, disulfide, in the tertiary structure of a protein. So one of the amino acids, cysteine, has an SH group, a thiol group, in its side chain, and when those 2 thiols get close to each other, they basically lose the hydrogens and the sulfurs bind together. They get reduction is loss of hydrogens. They get reduced and form a disulfide bond. A hormone is any chemical that's made in one place in the body and signals another place in the body. So some of our hormones are made from fats, from steroid hormones like testosterone and estrogen, but most of our hormones are actually made of protein. Hydrophobic effect. Hydrophobic interactions. These are ways that molecules react when they're afraid of water, hydrophobic. Inhibitor, I already described that. Inhibitors can be reversible or irreversible. So a reversible inhibitor binds using attractive forces, like London forces or hydrogen bonding, and can be removed. A non-reversible, an irreversible inhibitor is one that can't be reversed or removed because it covalently binds to the enzyme. Amino acids can be classified as either polar or nonpolar. They can be polar neutral or they can be polar charged. The charged ones are either positively charged and basic or negatively charged and acidic. So nonpolar amino acids are going to be ones that have nonpolar side chains, which will mostly be carbons and hydrogens, basically. Whereas polar amino acids are ones that have polar side chains. So an electron hog, like oxygen or nitrogen, are the two electron hogs that we see in amino acid side chains. Amino acids, by the way, are made up of carbon, hydrogen, oxygen, and nitrogen are the four elements in an amino acid. Let's see, I said peptide bonds already. Polypeptide is a string of amino acids that are linked together by many peptide bonds. So proteins can also be referred to as polypeptides because that's what a protein is. It's a bunch of amino acids linked together. There's, of course, the different levels of structure of a protein. The primary structure is just the sequence of amino acids. It's just the linear structure. The secondary structure then is either an alpha helix shape where that polypeptide either coils or it folds back and forth, and you can have sections of a polypeptide doing each. So one large polypeptide might have several alpha helices and several beta pleated sheets. The tertiary layer is the folding up of the protein into a three-dimensional shape. So the alpha helices and beta pleated sheets are in these different sections, and then the whole thing folds up into a three-dimensional shape. That's the tertiary structure. And then there's also quaternary structure. That's the fourth layer, and that's only for proteins that are made of multiple subunits. So if there's multiple subunits, so multiple folded polypeptides that come together, then a protein has quaternary structure. Protein and polypeptide are sort of synonymous terms. A protein is a polypeptide. Let's see. Salt bridge. A salt bridge is an ionic attraction between two charged side chains of amino acids in a protein. So salt, of course, is any ionic compound. So a salt bridge is sort of like an ionic attraction. It's like an ionic bond, except that it's not between two free-floating ions. It's between two ionically charged parts of a molecule. And the last one is substrate specificity. So substrate, remember, is anything that binds to an enzyme. And enzymes are very specific. They bind only specific things. So we call that substrate specificity. Now, some enzymes will have sort of stricter substrate specificity than others, which may be able to bind a range of similar looking molecules or similar chemicals. All right, I think that is all of the words for Chapter 10.